Okay, so I think we can go ahead and get started. I want to introduce Christian Zanetti, who is our Chief Technology Officer and a co-founder of Fluxion Biosciences. So I'll turn the presentation over to Christian, and we will get started with the webinar.
Hello, everyone. Thanks again for joining. My name is Christian Zanetti. I'm one of the founders of Fluxion Biosciences, and it's my pleasure to introduce our IsoFlux system for rare cell isolation. It's an exciting time at Fluxion. We've just introduced this product. The market launch was in November of last year. And this webinar will be focusing on uses of the system for mutational detection in circulating tumor cells. First of all, I'd like to start with an FDA disclaimer. Our system and the kits are listed as Class 1 devices with the Food and Drug Administration for in vitro diagnostic use. The listed intended use is for blood sample dilution and CTC enrichment. CTCs can be used then for further analysis. However, the content that we will discuss today highlights applications that are for research use only, so please keep that in mind. A brief introduction. I'd like to tell you why we think mutation detection in circulating tumor cells is important. This is one of the biomarkers that people look at for tumor tissue, and it is well accepted that alterations in the tumor cell genes are relevant to the progression of the disease. In fact, a lot of therapies that are now on the market are linked to specific mutations, and there are tests on tumor biopsies that are being used to direct treatment. However, tissue is a significant hurdle to fully utilizing this molecular approach. While the initial biopsy is often available, sometimes that is hard to come by, either due to the fact that tissue is in a place where it's hard to extract, but in all cases, monitoring the mutational status over time is not possible, either because the tumor has been excised or because there's just too much discomfort and risk to the patient. Given that, we believe that CTCs are a great format for tracking mutational status over time, but heretofore they have presented a lot of challenges, and we have developed our system to try to address some of these challenges and really develop validated workflows to be able to get this type of testing done in a consistent manner. So briefly, what we'll be talking about today, I'm going to start with an overview of the IsoFlux system, basically go through the design principles and the methodology we use to separate out circulating tumor cells from blood and other fluids. What we'll focus on afterwards is a representative workflow, a qPCR-based mutation assay. In particular, we looked at point mutations of the KRAS genes, and we'll present some analytical as well as clinical validation of this assay. Finally, we're going to talk about the extensibility to other genes, other targets, and some alternative approaches for looking at mutation detection. So this is what the system looks like. It's basically a benchtop system that starts from typically a blood draw sample and works to enrich and present the CTCs in a sample format that's ideally suited for molecular assay work. All right, so a little bit of background on CTCs. I'm sure this is well-known material for many of you, but basically tumor cells are shed from the primary tumor and they enter the blood circulation system. They've been used, they continue to be used as predictive and prognostic biomarkers in addition to tumor tissue. Again, the biggest advantages are ease of access. The blood draws can be taken many times during treatment, and that circumvents some of the problems with primary tissue availability. That can be used to monitor disease over time and look at progression and changing mutational status. The challenges that we've tried to address are have been there for quite a while, and they are basically due to the fact that the concentration of these cells in the bloodstream is very low. The challenge then is to recover enough CTCs in order to provide meaningful analysis that clinicians or clinical researchers will believe. Therefore, what we've really focused on is improving this recovery efficiency, and in particular, the number of cells and genetic material that can be extracted off chip. Again, the utility of circulating tumor cells is clear. One is in research during drug development. The genetic status in terms of mutations is being used to select patients for clinical trials and look at correlations between specific mutations and the efficacy of treatment. Again, in the clinic, these can also be used to stratify patients, to pick the most appropriate therapy, and in some cases, mutational status can be used as a companion diagnostic for certain drug therapies. So again, here are some of the challenges, and what we've done to address them will come next. So the key challenge, and what we've most focused on, is having enough CTCs per sample to accurately perform a downstream analysis. As you'll see in the rest of the presentation, the actual limits of detection are different for different assays. But generally, we believe that at least 10 circulating tumor cells are needed per sample in order to provide a meaningful analysis. Therefore, in order for this to get broadly adopted and have great utility, we need to have a system whereby a high % of the patients have measurable CTC levels. Otherwise, a lot of the testing will basically be a no-read type test, and it'll be hard to truly stratify patient populations. Also important is the sample format. We felt that we needed an appropriate sample to transfer to the downstream assay, like qPCR. Basically, we started out with a number of different requirements. We wanted to have the cells viable and intact. That's important for both RNA applications and imaging. Also, we wanted to have a minimum elution volume that reduces the carryover from the rest of the sample and the contaminating background. It also means that the eluted sample can go directly into a PCR reaction without further steps like centrifugation and reconcentration. All right, so as you know, there are a lot of approaches being developed to isolate circulating tumor cells from blood. There are size-based approaches, which have the advantage that they're label-free. However, this approach is non-selective, meaning you cannot distinguish different populations of cells based on specific markers. Affinity panning, meaning coating of surfaces with antibodies that preferentially bind tumor cells, has also been used. While cells can be immobilized to a surface, the challenge there has been to elute the cell sample and have it available for further analysis. elution process often leads to limited recovery and really large dead volumes in the sample. Again, cytometry has been another approach that's been used for really rare cells. That's been, again, hard to implement successfully because of the large volumes needed and the fact that there is some cell loss in the process. Now, immunomagnetic beads are actually the oldest approach that's been around. The basic approach is to have beads that are magnetic, paramagnetic, that have been functionalized with an antibody that can bind one or more surface markers for the tumor cells that are not present in white blood cells. Now, the advantages there are that it's a selective, highly selective approach. It can be tailored to different subpopulations. The challenge, however, has been the limited isolation efficiency, and this is where we really worked hard and employed our core competency in microfluidics to really improve the collection efficiency and the recovery efficiency of the cells off chip. So in conclusion, what we have designed the system to do is to really deliver CTC samples in an optimal format for molecular analysis. That means one has to have high CTC recovery in terms of presentations that present measurable genetic material. We have a system that can use user-defined capture antibodies. We basically provide a kit that contains beads that are IgG linker conjugated. Therefore, the user can add their own capture antibodies. We also provide a standard EpCAM conjugated bead. The sample format for downstream transfer was also an important feature of the system, and we really focused on having a low volume, highly concentrated sample that can be validated as a starting point for DNA, RNA, and protein analysis. So briefly, I'll talk about the instrument format. The instrument runs 4 samples simultaneously. Each sample contains typically the result of 1 blood draw. Because of the runtime, that's about 1 to 2 hours. You can run 4 experiments per day, so 16 samples per day is the throughput of the system. We use pneumatic pumping, so we have a cartridge where the biological fluids are contained in, and no part of the system ever comes in contact with the biological sample. That's really important in order to basically eliminate the possibility of carryover and contamination from sample to sample. The sample format, there's basically a single patient sample per cartridge, and the CTCs at the end of each run are automatically transferred to a tube that then gets taken further to qPCR or other assays. Here's the basic layout of the workflow. We have a sample prep step where the user provides density certification, basically a phi call, and removes the white blood cell fraction. That is about a 1-hour hands-off process. That includes the conjugation of the beads that get added to the Buffy code. Next comes running the sample. So this is the time spent inside the instrument. It's about a 40-minute flow time through our microfluidic cartridge. And finally, the cells are ready for retrieval in a collection tube. So here's the principle of separation and what sets this apart from other immunomagnetic approaches. We used a microfluidic approach, and what that allows us to do is to have a continuous flow system where the separation happens in a really well-defined region below permanent magnet. What that does is the magnetic field gradients are high and highly focused, and the cells pass through almost single file through this separation region. Therefore, each cell sees similar forces as it passes through. And the capture is done by basically shifting the balance between the drag force that's due to the fluid flow and the magnetic force that's due to the attached beads. The other thing that we've done is we've really enabled recovery off-chip. And the way we do that is by having the cells captured against this permanent magnet on a solid surface that we call the CellSpot. Basically, all of the cells are concentrated in a region that's about 1 millimeter across below the magnet. And a big challenge with microfluidic approaches previously, as I've alluded to, is that the cells don't just need to be captured and counted, they need to be efficiently recovered off chip, and there's often loss during that recovery step. The way we address that is by not having to remove the cells from the substrate. So what happens is this substrate that seals the top of the fluidic channel is removable. So at the end of the flow, the magnet along with the CellSpot substrate get lifted out of the fluidic flow path, and the cells are all contained in a hanging drop that has a dead volume of less than 5 microliters. Now, along with the resuspension volume that you see down here at the bottom of the tube, the cells can be recovered in a total volume that's less than 20 microliters. But of course, that volume can include your reaction mixture for the subsequent assay. So we feel like this is a very efficient way of taking the cellular material and going further to the molecular analysis. Our hope is that CTCs will play an important role in reshaping the landscape for cancer disease management. And the current standard is that the spread of the tumor is being assessed both at the primary site and throughout the body in order to look at changes in the disease state. What we think this will evolve to is looking at both the numbers of circulating CTCs over time, but more importantly, their mutational profiles and their gene expression profiles. We believe that that'll be more predictive and will be able to earlier give us an insight into the efficacy of the treatment regimen. So there are a number of points in the cellular pathway that people have chosen to interrogate in circulating tumor cells. There's, of course, DNA mutations and other DNA aberrations. There's RNA that can be interrogated for as a surrogate for gene expression, but also for mutations again. Finally, a lot of users choose to look at protein, either protein expression levels or generally localization of the protein, either inside or on the surface of the cell. So these methods are well vetted for primary tissue, and there are a number of tests that are being used either in the clinic or as companion diagnostics that use these various methods, including mutation detection, to prescribe treatment. Now, focusing in on mutations in cancer, there are a number of genes that are well known to be associated with the development of the disease. Here's a list of some of them, along with the frequencies for the different indications. I should point out that all of these genes are possible to interrogate using the workflow that we've developed. But what I'll be focusing on today and showing data from is our KRAS assay in particular. We just chose that gene because it's well known, has clinical utility in the primary tumor, and is a tumor that's fairly common to a large number of cancers. So what does KRAS mutation detection look like in colorectal cancer patients. And this here I'm referring to data from primary tumors. Basically, we know that 35 to 40% of these patients show some positive mutational status. This is being used to basically assess the possible response of the patient to Erbitux, which is an anti-EGFR therapy. The KRAS mutational tests have become part of the standard of care for CRC patients, and therefore we feel like being able to accurately determine the mutational status in circulating tumor cells will provide a way to map this out longitudinally, which is not possible with tissue. All right, so I'm briefly going to talk about the actual mutation detection workflow. So this is what happens after the sample comes out of our instrument in this tube, basically in this hanging drop that's 5 microliters. We suspend that in 20 microliters and then perform whole genome amplification. That is necessary so that the sample can then be divided and we can look at a panel of multiple mutations. In this case, we use the QIAGEN whole genome amplification kit. We believe that other methods are compatible with this approach as well. In order to develop the mutation detection assay, we work closely with Life Technologies, and we adopted their CAST qPCR kits. CAST has the advantage of being really sensitive and being fairly tolerant of background, and this is the reason that we picked this assay, and we've been optimizing the full workflow based on that. The first step in the process was to look at analytical validation and really determine the limits of detection for the point mutations that were of interest. So this is an example of some of the experiments we did. Here we just took gDNA equivalents from a cell line, MDA231, that's heterozygous for a KRAS mutation. And what you can see, as expected, is that the delta CT numbers increase as you go to lower and lower cell equivalents in the sample. Basically, for the KRAS assay, this delta CT measurement is what determines the mutational status, and it's interpreted based on a cutoff. The delta CT is a difference in CT between a mutant and a reference assay. And basically, the cutoff is established such that all wild-type samples are above the cutoff, and mutational positive samples are below the cutoff. So we have determined here that above 4 cell equivalents, we can accurately call out these mutations, and that's a very good level of sensitivity. The specificity, again, is quite good. We have all of the wild-type samples fall above the 21 CT cutoff. Now, if you load the sample with all mutant DNA, you get about a delta CT of 7, and that's what the green line is down here. Now, of course, what's really important is to look at a closer representation of what the actual CTC experiment will look like, and namely spiking in of this known mutation positive cell line into whole blood from a draw. So basically, for this experiment, we took the NDA cell line in different quantities and spiked it into a blood tube, then went through the full isolation protocol on the IsoFlux system and looked at the assay results. What I should mention is that for the previous slide, we used really high backgrounds in order to make sure that wasn't going to be an issue. So we used backgrounds of about 10,000 cell equivalents. Our backgrounds in the IsoFlux instrument for clinical samples are closer to 500 to 2,000 white blood cells. Therefore, you'll see here that the separation between the wild type and the mutation positive samples is actually better in our simulated IsoFlux samples. That's because likely because the background is lower. So you can see that here on the x-axis we have matched samples, the counting data for CTCs in matched samples, and on the y we have the delta CT, and we can very easily recognize mutations down to 9 cells in this case. But if you fit this curve, you can determine that 5 CTCs is really the limit of detection for this particular assay. So that is what we focused on here. Finally, I'd like to present some data from a clinical study on CRC patients that shows the detection of KRAS mutations using this workflow and this approach. So basically, each sample on the x-axis here is representative of a patient, and I have this data is not fully annotated because it's being prepared for publication, but there are a few take-home points that I can share with you. One is that all patients that were healthy controls were not positive for any mutations. Also, patients that showed zero CTC counts and matched samples did not come up with a mutation. So those are nice negative controls. Now, of the rest of the patients, we see sort of what we expect, which is that a significant fraction of the patients present mutations, and that matches literature values in terms of percentages. Now, many of these mutations were the same as the primary tumor mutation, so we had primary tumor status as well for these patients, but some of them also changed their mutational status. So in the time from the initial biopsy to the CTC draw, which was 2 months to 2 years, we found that, again, a significant fraction of patients had acquired mutations. Now, in no case have we observed patients with a positive mutational status in the biopsy, but that present wild type in the CTCs. So that all makes sense and is very encouraging for this workflow. Now, the real question that we wanted to answer, and what I alluded to in the beginning of the talk, is that we wanted to make sure that on an ongoing basis there was a significant majority of patients for which this test could be performed accurately and provide a real answer about the mutational status. And what it boils down to is figuring out what percentage of patients have enough circulating tumor cell material to go above the limit of detection of the assay, in this case 5 CTCs. And here I'm just showing you some samples that were analyzed from either whole blood for a number of different indications or leukapheresis. And if you take all this data and you sum it up, what you basically end up with in terms of patients above the assay limit of detection is something like this. So across all indications, we have greater than 75% of patients that present greater than 5 CTCs in enumeration experiments for a number of different starting conditions. So there are also a significant number of patients that have greater than 10 CTCs, and that's just a better sample. It's a sample that's far above the limit of detection, so we can call mutations with confidence. Now, we have worked with a number of collaborators, some of which have looked at other sample formats. And the idea here is to be able to cull white blood cells from a larger blood volume, that is, white blood cells including circulating tumor cells. And our collaborators at Bayer have used leukapheresis samples in order to improve the numbers of CTCs that are being recovered. And what you see here is for both breast and pancreatic patients. they have close to 100% of patients presenting over 10 CTCs per sample, and this is, the ability to do this is going to be very important for their patient stratification applications. So a little bit more about the Bayer study, and I should mention that the folks there have worked with our system for quite a while and are one of the first IsoFlux customers. What they really did is they made circulating tumor cells a real priority in their cancer biomarker program. And as part of that, they looked at a number of providers for capture technologies. So as you can see in this poster here, I think this is one of 2 posters that they've presented on this work. They looked at a number of different technologies and a number of different approaches and tried to optimize the sample and the recovery from their patients. This part of the poster basically shows the comparison between a multi-site study using the Veradex and results from the IsoFlux system. And as you can see, despite the fact that both systems use immunomagnetic separation, the improved isolation efficiency and off-chip recovery results in much higher numbers of cells. And these numbers of cells have translated, as expected, into better readouts for their downstream assays. In the case of Bayer, they're looking at protein expression and protein activation. So in the last part of the talk, I'd like to present some analytical data from a couple of other approaches to look at mutational status. Now, we believe that approaches like qPCR for a panel of point mutations that have known functions will be very important for both companion diagnostics and patient stratification in the clinic. However, early on in the discovery process, it's very important to be able to look more broadly at mutations, and therefore next generation sequencing is becoming important. So in this study, we worked with Assuragen, a CRO collaborator that was using the MySeq instrument to look at percent mutations in the samples that we obtained using the IsoFlux system. In this study, we looked at spike-in levels and cell recovery from blood samples, and we looked at the predicted percent mutants based on counts and compare that with the measured percentages, and they match very well. And this type of work is used to determine the limit of detection. For this particular assay, the limit of detection is going to be higher than for the KRAS qPCR. We estimate that's going to be about 30 to 40 cells per sample. Another approach that people are taking in order to really quantify the percent mutants in the sample is Droplet digital PCR. And in this, again, analytical study that uses a similar sample format that is MDA cells spiked into whole blood, we compare the unenriched results to the results from this instrument post IsoFlux enrichment. And I should point out we used the QX100 Droplet digital PCR system from Bio-Rad. And the real advantage of these types of systems is that you can get a quantitative number for the abundance of a rare mutation in the sample. So as you can see here, down to 10 cells that were spiked in, we can get a droplet count that is above the limit of detection. So what happens in these systems is that the DNA gets separated into a very large number of droplets, and only the mutation-positive droplets are fluorescently tagged and then counted downstream. So again, the purpose of this type of work is to determine an effective limit of detection. And in this case, when we spike in 10 of these cells, which means the recovery is likely 5 to 7 cells for this particular cell line, we get a positive signal, meaning the limit of detection is again likely around 5 CTCs per patient sample. Finally, I wanted to share with you some of the other assay validation work that's going on at Fluxion. I think that we have really focused on developing full workflows that start with a blood sample and end with an answer, and I think that's very important to provide our customers with these types of workflows. It's really the cell separation is very important, but what's even more important is the compatibility of that sample format with downstream assays. So you've seen a lot about our KRAS mutational test. We're also working on validating other targets using the same approach, things like EGFR and BRAF, and really all of the genes that are available in the Cas format are amenable to this approach. We have a validated enumeration test as well that's based on the usual 3 immunofluorescent markers. We're also working, as you saw, on next-gen sequencing approaches and FISH approaches, which continue to be important for tests like HER2 copy number. So in conclusion, I hope I show you some compelling data and convinced you that the IsoFlux system is been built and is ideally suited for mutational status determination for circulating tumor cells. I think this is due to the emphasis we've put on the high CTC recovery and the flexibility of the system with user-defined antibody cocktails being available. What we found for blood draws is that currently over 75% of patients present enough genetic material to make a positive determination of the mutational status, and that's been confirmed both analytically and in clinical samples where we have matched counts and we've detected mutations down to CTC levels below 10. Again, the transfer of the sample is very important. We have this unique CellSpot approach that enables a very low volume to be recovered along with the CTC sample, and I think that's also very important to the assay sensitivity. The resulting validated assays that are available are for a variety of different genes. All of the commonly found genetic mutations that I showed you in the previous slide in primary tissue are available in the Cas format, so therefore we can perform that using the IsoFlux and the workflow that's been developed. Finally, thank you for your attention and your time, and we hope to hear your questions and happy to answer them either publicly or in private. Thank you. So a couple of questions. One was about the quantification of the prevalence of the mutation. So there was a question about whether or not the qPCR method is quantitative. The answer there is the method is semi-quantitative. we can get an idea. As you saw, the CT number changes with the number of mutant copies, but it's not an exact count. To get exact quantification, one would have to use probably other methods like Droplet PCR or next-gen sequencing if one wanted the exact number of mutant copies. There was another question about Fluxion's involvement in downstream method development, and I think the question was whether or not we would help and participate in method development, and the short answer is yes. I mean, we have put a lot of effort into developing these assays that happen after the CTC sample is presented by the IsoFlux instrument. We think that's really important to demonstrate the utility of the system, and we have worked with a number of collaborators and are happy to work with others in developing new methods. There was another question about the modality used for CTC capture in these samples. Basically, a lot of the data you've seen uses the anti-EpCAM, the EpCAM antibody to select out the cells. We simply started working with EpCAM because there's a lot of existing data, and we wanted to provide sort of apples-to-apples comparisons with other capture technologies. We do have customers that are using different markers and combinations of markers using our sort of self-configurable bead system. However, that data is not publicly available yet. There's a question about the amplification kit that was used. We used a kit for QIAGEN. I'm happy to send you the full information privately. There was another question about multiplex mutational testing, whether that's possible with the system. The answer is yes. I mean, as you saw in these studies, we looked at actually a panel of 7 KRAS and 1 BRAF mutation. I think using this approach, one can go to tens of different point mutations. If there's a desire to go above that and look at large panels of mutations, probably next-gen sequencing would be most appropriate for that type of work. There was another question about the possibility of doing cell culture post separation in the IsoFlux system. I can tell you that the cells are viable, and we've done some work that indicates we can extract cells post separation and culture them using primary ex vivo cells. However, I will say that this is a challenging application, and that more work would need to be done to do that on a routine basis. And happy to talk to you more about our experiences there.